Good afternoon, class. We have our topic for today, which is genetics of animal infertility. Okay, let us have a review of our genetics, and we start with the chromosomes, which is a thread-like structure of nucleic acids and protein found in the nucleus of most living cells, carrying genetic information in the form of genes. So it could be a diploid or a haploid. A diploid means two members of each pair are present, like the sex chromosomes. We have either the XX or XY, or the autosome, any chromosome that is not sex-related or sex. On the other side, we have the haploid, which has only one member present instead of pairs. So such as the gametes, we have either the egg or the sperm. Okay, the genes determine one of or more characteristics or traits. And the autosomal and sex-linked concepts were already discussed previously. So we proceed with the dominant genes, which is shown in every litter or generation, or recessive, as it may be commonly absent or present but in low occurrence only in every litter or generation. And we have the concept of penetrance, which means that this particular trait can appear in every generation. So we can say that a dominant gene has high penetrance because it can appear in every generation. Recessive gene has low penetrance because it may skip a generation. So we know that some genes may only be evident on the grandchildren. Okay, it skips the parent level, so it can only be shown on the grandchildren level. So we have such variations that we see in humans and also in animals. Now, on the pathologies, we start with these chromosomal abnormalities. We have the first of two, numerical aberrations. We first discussed the mosaic, which has two or more populations of cells with different genotypes in one individual. So this can be tricky because it has developed from a single fertilized egg, but it has one or more populations of cells with different genotypes. Or a chimeria, so this is another entity. So a chimeria is a single organism composed of cells from different zygotes. So this can include possessing both female and male sex organs, blood cells of two blood types, or subtle variations in form. So animal chimerias are produced by the merger of multiple fertilized eggs. And now on the other side, we have the defects and the meiosis distribution. First, we have the euploids, and these are exact multiples of the haploid number of chromosomes. Examples, we have 2n equals 46 in humans. And the pathologies that we can get from these euploids could either be polyandry or polygyny. Polyandry has more than one male pronucleus or male sperm in an egg cell, and that would then equate to 3n instead of the usual 2n only. The other variation is what we call the polygyny, meaning there is only one male or one pronucleus or one cell, but has more than two females or two eggs, but still it equates to 3n. Okay, then we have the aneuploids, which is having an abnormal number of the chromosome. So coming from 2n equals 46, 2n minus 1 for monosomic, that will be 45, or 2n plus 1, that will be 47 for the trisomic. Okay. Next, we have structural abnormalities. So we have 6 variations here. We start with tandem fusion. So just portions of the chromosome would be would join as one. And then we have portions of the leg of a chromosome that is being removed. So we have deletion coming from, let's say here, the longer leg of the chromosome. Now the middle third is removed, resulting to a shorter leg of the chromosome. We can also have translocation, which could either be reciprocal or Robertsonian. But this just means that chromosomes interchange their legs or their arms, and it would be dictated by the location on where you have the translocation. So it could be either reciprocal or you have the Robertsonian. And then lastly, we have the inversion, which is in itself a segment of that leg of the chromosome is inverted. No? Okay. Furthermore, we can combine these chromosomal aberrations into two lesion abnormalities. So either we can have interchange, it could be dicentric, meaning at the middle part where you have your abnormalities. It can be centric ring, okay? At the middle, they would be joined together. Dicentric ring, okay? And then we have intra-arm interchange. Either we have interstitial deletion, so you remove a portion, or we have isochromatid deletion. One portion or one leg has been completely removed. And lastly, we have the discontinuity, meaning one leg has been removed permanently and not replaced at all. Okay, I have already summarized the different alteration types as shown in the flowchart earlier, together with its definition. And we have some examples here, particularly for the aneuploidy. So it means either you remove one from the 2N or you add one or two to the 2N. So we have the cat having 38 chromosomes. So for the aneuploidy, we have an additional N, which makes it 39. And instead of an XX for a female cat or an XY for a male cat, we have XXY. And we call this the Klinefelter-like syndrome, and it happens in cats. On another example, we have X. The horse has 64 chromosomes. So instead of getting another X or a Y, we have an O here. So the equation is incomplete, which makes it a 63. And we call this the Turner-like syndrome, which happens in mares. Okay. Secondly, we have the euploidy or the polyploidy. Again, inexact multiples of the haploid because we have an additional either an egg or a sperm. Okay. So from the 2N, it could be as simply as 3N or 4N or 5N because it does not end with just another female or a male. So it's more than one. So it could be 3N, 4N, or 5N. Okay. And we have coming from 178, it's now 180 because we have additional 2N, no? So from 178. And an example of the mosaic is translocation. So we have this either as an 60 normal or a 59, okay, which means that one chromosome has been removed, a Robertsonian translocation in the location in the loci 1,29. Okay, so this just means that for translocated aberrations, the bull can still have the normal 60 chromosomes or a 59 abnormal set of chromosomes, no? So either 60 or 59, what's important, at the level of the Robertsonian loci, it has been interchanged. Okay, so the location is always this I, 29. Okay, lastly, we have the chimeria. So we either have the normal 60 for cattle, XX, or 60 XY. And we can see this for free Martin heifer, which was born as a co-twin to a male bull. Ano yun? Male calf. Yeah. A bull calf. Okay, so it can either have the normal 60XX but still have an underdeveloped female reproductive tract, or a 60XY but with a female genotype. Okay. Another summary right here would be the basic types of structural chromosomal aberrations. So we have your deficiency, and instead of the Y for the boar, the capital Y, we have a small YT, and the location of where the aberration is. Okay, so that's your genetic aberration. A lost chromosomal segment. So it doesn't have the complete capital Y there. Or it can have a duplication. There's an extra chromosomal segment. An inversion. So same as what was shown earlier, no? Inversion, the segment is in a reverse sequence. Or then we have the translocation. Here in the bull, instead of having the 62N chromosomes, we have the Robertsonian translocation in dislocation in the loci, which happens in the bull. Or for the boar, we have 38, that's still the normal, but we have a translocation on this area of the loci. Okay? And that's for the bull, a boar. Okay, for the boar, now we have these external manifestations of these chromosomal abnormalities. So for our congenital malformations, this can be explained by having ovarian hypoplasia in mares because of the Turner syndrome, or testicular hypoplasia. So either the normal is XY, but instead of that, we have an additional XXY. So this affects the normal development of the testicles, leading now to testicular hypoplasia because of this extra Y because of that extra X chromosome at the start. Okay, for sterility and infertility, either this can be caused by your mosaic or your chimeria. Then we also have prenatal mortality, and that could be any defect, okay, that you can see that's leading to the incomplete maturity of the development of the fetus. That's why it's not strong as it is born. Then we have intersexuality. We have the true hermaphrodite, sumo hermaphrodite, or we have the free Martin. So the true and then the pseudo hermaphrodite will be explained later on, and these are genotype abnormalities. Okay, that can either be explained depending on the species. No? So if it's a cat, so we have the Klinefelter syndrome, and then if we have the cattle, then we have the aberration that was mentioned earlier. And then hybrids, okay, normally we have successful hybrids such as with the horse and then with the donkeys, but sometimes other species will not have successful hybrids because their chromosomes are not compatible. For example, we do not have any successful hybrids coming from cattle and buffalo because of the difference in the number of chromosomes, okay? Furthermore, these are the external manifestations resulting to infertility. So for testicular hypoplasia, which can be evident in the cattle, sheep, and the pig. So these are your chromosomal aberrations that would cause that, and then the respective karyotype. So for cattle, trisomy, meaning 1X is in addition to the normal. That's why the total number of the chromosomes is now 61. This should be male, but then we have the extra X here, and that leads to testicular hypoplasia. So same as how I explained it earlier. So for chimeria, the karyotype may be normal, either as an XX or an XY. Well, if it's a male and it has an XX, so it just means it has that extra zygote Okay, that makes it abnormal. Okay, for the sheep, we have also trisomy, and for the pig, we also have trisomy, which has an additional one from the normal 54, an additional one from the normal 38. So we have this additional X chromosome, leading to the testicular hypoplasia. For ovarian hypoplasia, which can be more on the horse as the Turner syndrome, monosomy instead of the normal 2N. So we have 2N minus 1 that leads to 63. For trisomy, additional X, 2L plus 1 that leads to 65, and XXX, okay? And for the cattle, we also have trisomy. For the female, that's additional X, okay? So anything that is abnormal, anything in excess of the normal, be it an additional or a subtraction to the normal set of chromosomes, will lead to abnormalities for both the male and the female. So when we're talking about genetics, it should be exactly the same all the time. Any slight difference, even to like if it's in numerical form, even to the 0.001% abnormal, it would really lead to any abnormal manifestations already. Okay. For any repeat breeders, which we usually see in cattle, if you want to investigate further, if it's not because of management, hormones, nutrition, etc., on a deeper sense, we can look at it genetically. And it can be because of it is a mosaic or it's a mixoploid. Okay. And again, moreover, for infertility, it can be attributed to Robertsonian translocation in this level. Okay. And then for embryonic mortality in pigs, it could be because of this translocation. Okay. At this level of the loci. Okay. So this T parenthesis 7Q minus semicolon 11Q plus, this is the exact location where you have the translocation, okay? And these are some that can be transmitted vertically into generations. First, we discuss the morphologic defects, and you see a lot of these cryptorchidism, be it unilateral or bilateral, in our stallions and boars. That is why if we see this in one of our animals or patients, it's very important not only to do the cryptorchidism, but cryptorchidectomy, but also do the castration on the normally descended testicle if it is a unilateral cryptorchidism. Okay, so it's very important not to spread the genetic line of these cryptorchid animals, because it is a dominant gene that can be transmitted vertically. Okay, moreover, we have gonadal hypoplasia, which is evident in a particular breed of cattle, the Swedish Highland cattle. So this is a beef type cattle, not dairy. Okay, we can also experience some spermatozoal defects. And for duct abnormalities, either on the male side or the Wolffian duct, or the female side, or in the Mullerian duct, there is underdevelopment of this certain ducts. That's why it can lead to fremartinism, or we have this hypoplasia. Okay. For functional disorders, the external signs would be either cystic ovaries, leading to nymphomania, or lack of libido in terms of the male counterparts. To summarize again, we have this examples of inherited abnormalities of the male and female reproductive system. So from the cryptorchidism, this is sex trait, and because of the undescended testicle, there is disturbance in the spermatogenesis. Okay, the normal descended testicle is not affected, only the cryptorchid one. Okay, if unilateral. Then for the gonadal hypoplasia, meaning the vulva, or the penis is not developed properly. So it could be because of a recessive autosomal gene. And since it is recessive, no, it has incomplete penetrance. So they're directly related. And because of this hypoplasia, scrotum or the penis, there is then sterility or infertility. So the white heifer syndrome that can be seen in cows is recessive genes for white coats. So for animals that are purely white, they can have this white heifer syndrome, wherein the female reproductive tract did not develop properly. Let's say one segment or one uterine horn is not normally large. It is rudimentary, okay, or one ovary is missing. So these are your variations for your white heifer syndrome. And this could lead to sterility, especially if both left and right sides of the uterus are affected. Next, we have the male pseudohermaphrodite, which can be exhibited by the goat, pigs, the goat and pigs. And this could be inherited through a dominant autosomal gene for poled horn. Okay, so this is one trait that we have to be particular with when we're dealing with poled small ruminants, no, poled sheep, no, and then poled well, most goats are really without horns, but sometimes we have goat with horns. So some polled goats as well. And this could be and this could lead to sterility, meaning okay, they will not be able to produce good quality sperm to be able to expand their genetic line. Okay, then we also have some cystic ovaries, which are very common in cows. And I have limited explanation or information that I posted in our Google Classroom earlier. Okay, for the cystic ovaries, and it could be due to nutrition, genetics, okay, stress. But relating to genetics, okay, it could then lead to either an estrus, but mostly to nymphomania. Okay, this is the white heifer disease that I've been mentioning earlier, and this gene arrests the development of the Mullerian ducts. Okay, and you see this beef cattle that is purely white. Okay, it's a short horn breed or a pole breed that is purely white, and we can mostly see this in cold countries, in temperate countries. So figure 10 is showing us the class 2, meaning only one side or one horn of the uterus is present and one is rudimentary. Okay, for class 1, we have segmental aplasia of the Mullerian ducts, not not the entirety of the uterine horns, okay, is problematic. Only a portion, let's say the fallopian tube or the uterine horn, no, either of those two. And then for class 3, we have imperforated hymen. So then it's difficult for the penetration of the penis needed for breeding or reproduction. Okay, so we have figure 9 here as an example of class 1. We have region of the uterus and cervix with cystic dilatation of the uterine horn. Now, on the male part, we have different abnormalities of the sperm, and we have the respective description leading to either infertility or sterility. So let's have an example here of an sperm, and we have a picture. The thickening of the acrosome, which is the cap of the sperm, and that leads to sterility. Okay, sterility is the permanent, okay, inability to procreate. And then another example, we have, let's say, corkscrew, and we have a picture here of the tail that is twisted, and this leads to infertility. And now on the last part, we discussed the hermaphroditism. We have two major types. We have the true and then the pseudo. For the true hermaphrodite, we have either bilateral or lateral. Four gonads, meaning the male and female uterine horns. Left and right uterine horns. And then for the male, we have the left and right testicles. Okay, for the lateral, we have either two of the uterine horns or two of the testicles, and unilateral, which has a low incidence. We have either two testicles and one of the uterine horn, and vice versa. Okay. Then for the pseudo, it can be male or female pseudo, meaning for the female, no, it could be the animal is female inside but not showing, but showing otherwise outside. Okay, and then vice versa. So we have high occurrence of pseudo-hermaphroditism in our animals. Okay, I have a case here, and her name is Tomboy. She is a doe, so a goat, and she has not been pregnant, and also with poor parang libido and poor estrus, poor estrus. And you can see that the face exhibits more of a buck, okay, rather than of a doe, kasi hindi yung kanyang mukha. Although this is a mixed breed already, and ang Lunubian probably with a native, because it's already bigger than the size of a native, and its coat is indicative that of an Anglo-Nubian. Okay, but see the thickening of the forehead, which is not indicated for female does. So nawalan siya ng parang feminine sense in the face and in the body. Okay, so I did not investigate further rather than just looking on the physicality and on the history of Tomboy, the patient. Okay, there's no means for me to really investigate genetically, but we can say no that she is a hermaphrodite because she has not bred successfully. So she may have excessive male reproductive organs inside her. Okay, for true hermaphrodite abnormalities in the goat, the karyotype would either show as your normal 60XX or it could be 60XY. So the gonads that can be present would be both the ovaries, okay, and then the testicles. The external genitalia would be female and the sexual behavior would be female. Okay, that would be the same for the pigs. It's just that it has several variations of its karyotype. Okay, so it's either 38XX or 38XY, or it can be as varied as 39XY or 37XO or 38XX, 38XY. Okay, so there are several variations of the true hermaphrodite in pigs. For the X, we have the Turner syndrome again. And the sexual behavior as a difference to the two previous domestic animals mentioned, it is that it has a lack of the male behavior. So it's really more of the female, but it has the two testicles inside, okay? So, and for the cattle, and it has the ovary and ovipositis, and the external genitalia would have the penis, and the sexual behavior would indicate much more of the male rather than the female. Okay. So we have the phenotypic masculine or feminine, depending on the dominating gonads. We have enlarged clitoris, but there's no differentiation on the cervix. So the cervix may be the same. That we see that has abnormalities would be on the uterine horn or on the ovaries, and then the presence of the rudimentary testes. Okay. And we have one of the clues that we can look at is on the enlarged clitoris, but small underdeveloped vulva. So you can also see this, aside from hermaphrodites, you can also see the underdeveloped vulva on free Martinism, okay? But that's on another category, no? Because free Martin is a chimera. It's an example of a chimera. Okay, so both genital tracts are present but are undeveloped, no? Both are undeveloped for your true hermaphrodites, and they would come out as infertile. Okay, looking at the histopathology of this through hermaphrodite, we have both the cells of the testes together with the cells of the ovary. Okay, and we have here the ovaries, okay, and then the small testicles right here. Okay, right ovary here, and then B is the bottom testes, and then C is the right upper testes, okay? But in here, we can mostly see just one of the testes. Here, it's not just very, but it seems this is the other testes, and the middle is the rudimentary ovaries. So the urinary bladder is not affected, the uterus is also, the cervix is not affected, but mostly it's on ovaries. Okay, now for pseudohermaphrodites, instead of having still female sexual behavior, we now have male sexual behavior. So that's the main difference between the true hermaphrodites for the goat and then the pig. Okay, because of the presence more of the testicles inside, but externally they look females. For the horses, no, we don't have the Turner syndrome because we mostly look at it on the male side. And then we have the testicles, but they are inside, like they're cryptorchids, and they have enlarged clitoris, okay? Enlarged clitoris, but still the sexual behavior would be exhibited as male. Okay, for the cattle, we have this cryptorchidism or abdominal testicles, and the presence of enlarged clitoris, and presence of small rudimentary vulva, and but the sexual orientation, I mean the sexual behavior being exhibited is male, or not exhibiting estrus. Okay, so it means that it means it means when I say male sexual behavior exhibited, it means that there is some aggressiveness and there is increased libido time, all the time, okay? Because it's not indicated by the estrous cycle. Okay, so it's the same for the sheep. So we have this a similarity for the cattle and the sheep. And then now we have here free Martin, Chimera, which can also be categorized as sumo hermaphrodites because of their small rudimentary vulva but enlarged clitoris, and they're not exhibiting the feminine external characteristics of our cattle, but they are steer-like, meaning same as what was given to us by Tomboy. The forehead is skin is thickened, no, and for other cattle, no, you can see that they have enormous horns, okay, and they're masculine, no, and they look big, no, so that's your uh male external signs. Okay. For goats, we see a lot of pseudohermaphrodites. They are polled. They could either be male or feminine in appearance, and the external genitalia is more like this. So see, the one that's where I'm pointing, we have a very small vulva, okay, and the testicles cannot really be seen outside because they are inside the ovaries. And then if it is a male and a male pseudohermaphrodite, the penis is not entirely developed, so it will be short and it may be bent upwards, okay? So it's really not meant for breeding. Okay, now we have this gross pathology in pseudohermaphrodites. We have the testes, okay? And then, but we do not have the rudimentary ovaries, okay? Just the testicles right here. And then we have the presence of underdeveloped penis with a bent, okay, end. Okay, and the masculine type of male pseudo hermaphrodite is also the same. And we have this very small vulva. It has no scrotum, abnormally short. Okay, same as what was shown earlier. And lastly, we have the reproductive tract of said male pseudohermaphrodite. And we see that there is here the ovary, wait. So this is the left horn, and this is your uterus, okay, but it's not very entirely developed. And then we have an ovary right here. Okay, so this is abnormal because it came from male. So it should not have this female reproductive tract inside. Okay? So that ends our meeting and our lecture for today. Do you have any questions? If you don't have any at the moment, you can send them to me via my email, and then I will just post the question together with the answers in our Google Classroom.